student of the Department of Medical Biochemistry. Topic of my lecture is Introduction to Metabolism, Specific and General Pathway of Carbohydrates, Lipids and Protein Metabolism, Oxidative Decarboxylation of Pyruvate, Krebs cycle. Metabolism, the entire network of chemical reaction carried out by living cell. Metabolism also includes coordination, regulation, and energy requirement. Metabolites, small molecule intermediates in the degradation and synthesis of polymers. Most organisms use the same general pathway for extraction and utilization of energy. All living organisms are divided into two major classes, autotrophs and heterotrophs. Autotrophs can use atmospheric carbon dioxide as sole sources of carbon for the synthesis of macromolecules. Autotrophs use the sun energy for biosynthetic purpose. Heterotrophs obtain energy by ingesting complex carbon-containing compounds. Heterotrophs are divided into aerobes and anaerobes. Common features of organisms. Organisms or cells maintain specific internal concentration of inorganic ions, metabolites, and enzymes. The second one, organisms extract energy from external sources to drive energy-consuming reactions. Organisms grow and reproduce according to instruction encoded in the genetic material. Organisms respond to environmental influences. Cells are not static, and cells components are continually synthesized and degraded. For example, undergo turnover. A sequence of reactions that have a specific purpose, for example, degradation of glucose, synthesis of fatty acids, is called metabolic pathway. Metabolic pathway may be three types: A. Linear, cyclic, and spiral. For example, linear is 3-phosphoglycerate converted to serine. Cyclic, for example, Krebs cycle, and spiral pathway, for example, fatty acids biosynthesis. Metabolic pathways can be dropped into two paths: catabolism and anabolism. Catabolic reactions degrade molecules to create smaller molecules and energy. Anabolic reactions synthesize molecules for cells, maintains growth and reproductions. Catabolism is characterized by oxidation reaction and by release of free energy which is transformed to ATP. Anabolism is characterized by reduction reaction and by utilization of energy accumulated in ATP molecules. Catabolism and anabolism are tightly linked together by their coordinated energy requirements. Catabolic process release the energy from food and collect it in the ATP. Anabolic process use free energy stored in ATP to perform work. Anabolism and catabolism are coupled by energy. For example, biomolecules which degrade in catabolism to building block and produce energy. This building block used in anabolism and synthesis new biomolecules and usage energy. Levels of metabolism regulation. There are 5 levels of metabolism regulation. First of all, nervous system, endocrine system, like hormones. The third one, integration between organs, for example, cycle Cori, cells membrane level, and the fifth last at molecular level. Stages of metabolism. Catabolism divided by 3 stages. Stage number 1, breakdown of macromolecules, protein, carbohydrates, and lipids to respective building blocks. Stage number 2, amino acids, fatty acids, and glucose are oxidized to common metabolite acetyl coenzyme A. Stage number 3, acetyl coenzyme A is oxidized to in citric acid cycle to carbon dioxide, and water. As a result, a reduced cofactor NADH2 and FADH2 are formed which give up their electrons. Electrons are transported via the tissue respiration chain and released energy is coupled directly to ATP synthesis. In human body, big macromolecules degrade to monomers, proteins to amino acids, carbohydrates, glycogen and starch to glucose, lipids to acetylglycerol to glycerol and fatty acids. Is the first stages of catabolism is specific stages. After this, amino acids, glucose, glycerol and fatty acids converted to common metabolite acetyl-CoA is the second stages of catabolism. Its specific stages too. The third stages of catabolism as conversion of acetyl-CoA and producing energy in citric acids and during the oxidative phosphorylation, producing energy and formed carbon dioxide and water. Is the third stages of catabolism is common stages to all macromolecules in human body. Catabolism is characterized by convergence of three major routes toward a final common pathway. Different proteins, fats, and carbohydrates enter the same pathway, tricarbonic acid cycle. Anabolism can also divide into stages. However, the anabolic pathways are characterized by divergence. Monosaccharides synthesis begin with carbon dioxide, oxaloacetate, pyruvate or lactate. Amino acids are synthesized from acetyl coenzyme A, pyruvate or keto acids of Krebs cycle. Fatty acids are constructed from acetyl coenzyme A. On the next stages, monosaccharides, amino acids, and fatty acids are used for the synthesis of polysaccharides, proteins, and fats. Experimental method for studying metabolism. Add labeled substrate to tissue cells and follow emergence of intermediate. Use sensitive isotopic traces. Verify pathway step in vitro by using isolated enzyme and substrates. Study the mutation of genetic associated with the production of defective enzymes. Use metabolic inhibitors to identify individual steps and sequence of enzyme in a pathway. Oxidative decarboxylation of pyruvate. The fate of glucose molecule in the cell. Glucose during glycolysis converted to pyruvate. Pyruvate is metabolic bridge between glucose and acetyl-CoA. Oxidative decarboxylation of pyruvate. Only about 7% of the total potential energy present in glucose is released by glycolysis. Glycolysis is preliminary phase preparing glucose for entry into aerobic metabolism. Pyruvate formed in the aerobic conditions undergoes conversion to acetyl-CoA by pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex is bridge between glycolysis and aerobic metabolism, citric acid cycle. Pyruvate dehydrogenase complex and enzymes of citric acid cycle are located in the matrix of mitochondria. Pyruvate dehydrogenase complex is a multi-enzyme complex consists of 3 enzymes and 5 coenzymes and other proteins. Enzyme of pyruvate dehydrogenase complex. Pyruvate dehydrogenase. The second, dehydrolipyl acetyltransferase. The third, dehydrolipyl dehydrogenase. Coenzymes TPP, thiamine pyrophosphate, lipoamide, IHS, CoA, FAD, and NAD. TPP is a prosthetic group of pyruvate dehydrogenase. Lipoamide is prosthetic group of dehydrolipyl acetyltransferase, and FAD is a prosthetic group of dehydrolipyl dehydrogenase. The building block of DPP is vitamin B1, thiamine. The building block of NAD, vitamin B5, nicotinamide. 2. FAD, vitamin B2, riboflavin. HA squared, vitamin B5, pantothenic acid. And lipoamide is lipolic acid. Pyruvate dehydrogenase complex is a classic example of multi-enzyme complex. The oxidative decarboxylation of pyruvate catalyzed by pyruvate dehydrogenase complex occurs in 5 steps. Aerobic cells use a metabolic wheel, the citric acid cycle, to generate energy by acetyl-CoA oxidation. The citric acid cycle is the final common pathway for the oxidation of fuel molecule, amino acids, fatty acids, and carbohydrates. Most fuel molecules enter the cycle as acetyl coenzyme A. Another name of citric acid cycle, tricarbonic acid cycle or Krebs cycle. In eukaryotes, the reaction of Krebs cycle takes place inside mitochondria. Of the citric acid cycle, 4-carbon oxaloacetate condenses with 2-carbon acetyl units to yield a 6-carbon citrate. As an isomer of citrate is oxidatively decarboxylate and 5-carbon alpha-ketoglycerate is formed. Alpha-ketoglycerate is oxidatively decarboxylate to yield a 4-carbon succinate. Oxaloacetate is then regenerated from succinate. 2-carbon atoms, acetyl-CoA, enter the cycle and 2 carbon atoms leave the cycle in the form of 2 molecules of carbon dioxide. 3 hydride ions, 6 electrons, and are transferred to the 3 molecules of NAD, and 1 pair of hydrogen atoms, 2 electrons, is transferred to the 1 molecule of FAD. The function of citric acid cycle is the harvesting of high energy electrons from acetyl-CoA. The first reaction of Krebs cycle is citrate synthetase reaction. Citrate formed from acetyl-CoA and oxaloacetate. Addition of 2 carbon units acetyl to the keto double bonds of 4 carbon acids oxaloacetate to produce 6 carbon compound citrate. The second reaction of citric acid cycle is aconitase reaction. Elimination of water from citrate to form double bonds of cis aconitase. Stereospecific addition of water to cis aconitase to form isocitrate. The third reaction of Krebs cycle, isocitrate dehydrogenase. Oxidative decarboxylation of isocitrate to alpha-ketoglycerate is metabolically irreversible reaction, one of the 4 oxidation reduction reaction of cycle. Hydride ions from second position of isocitrate is transferred to NAD to formed NADH. Oxaloacetate is decarboxylate to alpha-ketoglycerate. This reaction catalyzed by isocitrate dehydrogenase. The fourth reaction of Krebs cycles is alpha-ketoglycerate dehydrogenase reaction. This reaction catalyzed by alpha-ketoglycerate dehydrogenase complex. This complex similar to pyruvate dehydrogenase complex. This complex presents 3 enzymes: alpha-ketoglycerate dehydrogenase with prosthetic group TPP, dehydrolipyl succinyl transferase with flexible lipoamide prosthetic group, and dehydrolipyl dehydrogenase with FAD. Alpha-ketoglycerate converted to succinyl-CoA, catalyzed by alpha-ketoglycerate dehydrogenase complex, and 1 second molecule of NADH formed in this reaction. The next reaction of Krebs cycle is succinyl-CoA synthetase reaction. From this reaction, free energy of diester bonds of succinyl-CoA is converted as GTP or ATP in higher animals. This reaction will start substrate level phosphorylation reaction and form molecule of ATP. The GTP formed in this reaction then donate its terminal phosphate group to ATP and formed ATP. The next reaction of Krebs cycle is succinate dehydrogenase reaction. In this reaction take part complex of succinate dehydrogenase. Its complex of several polypeptides of FAD prosthetic group and aryl cluster embedded in the inner of mitochondrial membrane. Electrons are transferred from succinate to FAD and then to ubiquinone in electron transport chain. From this reaction formed fumarate. The next reaction of Krebs cycle is fumarate reaction. Stereospecific trans-addition of water to the double bond of fumarate to formed L-malate. Only L-isomer of malate is formed. The last reaction of Krebs cycle is oxidation of malate to oxaloacetate. In the last reaction of the cyclic, the NAD linkage malate dehydrogenase catalyzes the oxidation of malate to oxaloacetate. As a result of the citric acid cycle, 2 carbon atoms enter the cycle in the form of acetyl-CoA. 2 carbon atoms leave the cycle in the form of carbon dioxide. 4 pair of hydrogen atom leaves the cycle in the full oxidation reaction. 3 molecules of NAD and 1 molecule of FAD are reduced. 1 molecule of ATP is formed. 2 molecules of water are consumed. Energetic balance of citric acid cycle. Producing 9 molecules of ATP are produced during oxidative phosphorylation. 1 molecule of ATP is directly formed in the citric acid cycle substrate level phosphorylation reaction. And energetic balance of total citric acid cycle 10 molecule ATP. Function of the citric acid cycle. First, integration of metabolism. The citric acid cycle is amphibolic, both catabolism and anabolism. The cycle is involved in the aerobic catabolism of carbohydrates, lipids, and amino acids. Intermediates in the cycle are starting points for many anabolic reactions. The second yields energy in the form of ATP, yields reducing power of the form of NADH2 and FADH2. Regulation of the citric acid cycle. 3 ways of regulation: allosteric modulators, covalent modification of cycle enzyme, supplier of acetyl coenzyme A. 3 enzymes have regulatory properties: citrate synthetase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase complex. Krebs cycle is a source of biosynthetic precursors. The citric acid cycle provides intermediates for biosynthesis. For example, oxaloacetate is one of metabolite of Krebs cycle converted to phosphoenol pyruvate and phosphoenol pyruvate converted to glycolysis during the gluconeogenesis, or another variant, oxaloacetate converted to aspartate and aspartate converted to pyrimidines and purines and another amino acids. Well, we finish. Thank you for your attention.